Hi, today we will be doing a fetal pig dissection. We've bought a pig off of Amazon here and we're going to first unbox it. So, some towels, and we have the pig. So this is a 14-inch pig. It's in the bag right now. It comes from a company named the Anatomy Lab. We're going to be doing the dissection in a 12-inch pan here. So it kind of barely fits. And here you can see it. Alright, so now we've taken the pig out of the bag. We've drained it a bit. You can see externally this is what the pig looks like. And we're just putting it in the pan. So first I want to kind of introduce the dissection we're doing. So as I said, this is a fetal pig dissection. So before any dissection, we should kind of make sure we know the orientation terms that we're going to use. So four major ones I'm probably going to use during the dissection are going to be anterior, posterior, ventral, and then dorsal. Anterior is referring to nearer to the head, so in this direction. Posterior is the other way, so kind of towards this direction. Ventral is the belly, so over here. And then dorsal is kind of the back. So when we talk about various structures on the pig, we're going to use these four terms. For example, the head is more anterior to the belly. The belly is more ventral than the back. We're going to use kind of simple equipment to do this dissection. First one we have are scissors. These scissors will be used to kind of snip away at tissue. Also, if we don't need a scalpel, it's more preferred to use the scissors instead. We'll also be using the scalpel. The scalpel is more sharp, so exercise caution when you're using the scalpel so you don't like cut your hand. And also we have a probe. The probe is to kind of point out or trace the direction of various organs. And also we should be exhibiting basic PPE, so like gloves or goggles if you have it. And if you have a lab coat, that would be even better. This is the external anatomy of the pig. Notice that it's kind of squished because it's been sitting in a bag for a while. So when we actually start the dissection, we want this pig to be ventral side up. And what that means is that the pig will be kind of in this direction. And then we're also going to have to kind of spread the limbs a bit. Before we start the dissection, though, we might want to point out some external features. This way, you can see that this is the head of the pig. These are the nostrils, and then you have the tongue. You can maybe see it. Well, the tongue has kind of these sensory papilla. This is how it tastes. And then we have the eyelids. Inside, we might be able to see the eye. And then we have the ears, also known as the auricles. And then there are, two of the ears. And then when we go down here, you have the forelimbs. The forelimbs have two joints. The first one is kind of the elbow, which is over here. And then you have wrists. So again, elbow, let's try the other side. This one's not very visible, but the elbow is around here. And then the wrist is around there. And then we also have the hind limbs. Hind limbs also have these joints. Here is the ankle. So kind of this bulge. And then the knee is up here. That bulge. And then again on the other side, we have ankle. And then knee. Around here. To determine the gender of the pig, we need to kind of look at the urogenital openings, figure out where they are. Oh, the tail's kind of snapped on this one. But to do so, you look posterior region of the pig. umbilical cord here, and you kind of look for a hole. Its position will determine what gender this pig is. So if the urogenital hole is just anterior to the anus, so around this area, then this is going to be the female pig. And if the urogenital hole is just posterior to the umbilical cord, so around this area, then that's going to be a male pig. This seems to be a male pig. If you look just posterior to the umbilical cord, you might notice this hole. So that's the urogenital hole. This is what determines that the gender of this pig is a male pig. And so if we kind of move posteriorly, there's also kind of a bulge over here. That means this is probably the scrotum. Yeah, then we can begin preparing the pig for the dissection. So an easy way of kind of keeping these limbs open is to use rubber bands. So kind of what we would do is get 2 rubber bands and then tie knot. So like that. So now you have kind of 2 openings, and then you try to secure one end of it. So having just 2 rubber bands tied together was not big enough because our pan is kind of wide, and so it wouldn't be able to kind of hold the legs in place. So what we did is we took 2 of them, we tied it together, and now we're going to secure the forelimbs. So what you want to do is loop one of the rubber bands around one of the limbs, then move the rest of the rubber band under the pan, and then loop it over. And then loop the other side around the other limb. Yeah. So then you're able to kind of pull the 2 limbs outward, and it will hopefully stay in place. You might notice that it's kind of going to create a band around the legs. That should be fine. Then you want to do the same thing on the other side. So have another rubber band. So now we're going to cut open the pig. So the first step we want to do is to cut a line down here, and we'd use a scalpel for this. So with the scalpel, we want to cut kind of superficially in the beginning, as to not cut too deep and then damage the internal organs. So yeah, you might want to flatten out the belly of the pig a bit so it's kind of easier to see what you're cutting. And yeah, we will begin. Again, when using a scalpel, make sure that you're being cautious. And you'll want to cut down until you kind of hit the nipples here. And you can kind of see the cut. There will be a rib cage under it, so you can kind of adjust the cut or use scissors later on. Yeah, so that's the first cut that you'll make. The next cut, let's put this down. Next cut will be kind of in a oval-like shape. It will be surrounding the nipples, and then we'll cut straight down after that. So just maybe move up the pig on mine. Yeah, you can move the umbilical cord out of the way, and then continue to cut. Again, we'll be using scalpels for this step. So that's one side of the cut, and then you want to kind of repeat the step and mirror it across the middle line. So again, cut in a kind of circular or oval shape around of the nipples, and then cut straight down afterwards. Okay, so now we have the first few cuts done. And since it might not be deep enough, like mine currently is, we can use scissors to kind of trace along the cuts, and then make sure you don't cut like straight down, but cut kind of level to the ground. And this way we'll be able to kind of cut away the muscles or the rib cage that might still be intact. So we'll get scissors. Around here, where my scissors currently are, this is kind of where the rib cage is, so it feels a lot harder. Just make sure not to cut like deep straight down, because under is going to be various organs such as the heart, liver, etc. Okay, we will want to make 2 cuts around here. So kind of what the shape you want to make is an inverted V around the base of the forelimbs. So you'll use your scalpel to do this. Just make an incision. Again, be careful with using scalpel. Make sure your blade is tight. And make sure your fingers are not in the way. Alright, and then after that you'll want to kind of do a similar cut around the hind limbs. Okay, so what we want to do now is we want to expose the internal organs. So first what we would do is we would take the umbilical cord and kind of pull up on this piece that we've cut out earlier. So you should be able to just pull back. If there's any like connective tissues, you can take your scissors and cut that out. Okay. So this cut was not deep enough. Make it a bit deeper. Again, as I kind of noted earlier, if you don't need to use scalpels, use the scissors. Alright, so you can kind of see some of the organs already. Alright, and then the next step is we take these 2 flaps and we kind of pull them open like a book. Okay, so now you can kind of see the internal organs. You notice there's a lot of liquid. Yeah. So if you want to get rid of this liquid, you can get a paper towel, kind of just dab up the wet areas. Okay, so here is what you should see when you pull open the flaps. You can see a bunch of organs, for example, just like some notable ones. The biggest organ you should see, that's the liver. And then you can also kind of see the triangles that are the lungs, the heart. So in the more posterior region, this is the digestive system. Digestive system, again, has the liver on the top. And then under the liver, let's see if we can find it, there's multiple lobes to the liver, so if you pull each one up. If you look kind of for this small bean-like structure, that's called the gallbladder. The gallbladder stores the excess bile. Doesn't seem like I can really find it, but we can look deeper when we actually take out these organs. So again, liver, this flap here, this is the stomach. And then next to the stomach, attached to it should be the spleen. Spleen will kind of filter blood and then create white blood cells. So this is the spleen. This portion that's attached to the stomach. And then under that you have the intestines, which you can see here. And then the rectum is kind of more posterior than intestines. So this kind of portion is the digestive system. More anterior to it is the respiratory system. So you can kind of see the diaphragm is slightly detached at this point, probably because I cut part of it. On top of that, there is the lungs, the triangular structure. There's two lungs, one here, the other one's on the other side. It's kind of blocked with here. And then in the middle of that is the heart for cardiovascular, but heart is here. On top of the heart, there should also be the thymus. It's kind of not visible. There's more glands structures up here, which we will kind of cut further later on. But yeah, this is the general overview of the organs you might see when you open up the pig. Kind of on the dorsal side, more dorsal to the two visible systems here, you also have the urinary system, which includes like the kidneys and then the genitals. So kind of to access that, we can take out the digestive system. And to do so, you would first kind of pull on the liver and disconnect it from the rest of the system. So here is the digestive system. After we've taken it out, you can still see the livers on top. The stomach, spleen should be next, yeah, here. And then we also have the intestines. Yep, so now we can put this to the side. So if you have another container, just put it to the side. Okay, so now we've removed the digestive system and we can kind of see parts of the urinary system. So I had to kind of clean out some of the liquid here. But you might notice the kidneys. There's two of them, right? Bean-like structures. One of them's here. The other is on the other side. And right now they're covered by some membrane, but we can try taking them out. So, yeah. So you can see the kidneys, also what seems to have been in probably the rectum. Yeah. On top of the kidneys, there should be the gland called the adrenal gland. Returning to kind of the respiratory or cardiovascular system. There is more structures up here, so we can continue cutting up. Cutting up on this area, we kind of can see that I've cut through, but part of it's still intact. The thymus, if it's intact, kind of has a U-like structure, which was seen in the slides that I made. But looks like this is the outline of the thymus. You can kind of notice the two ends of it. Okay, and the thymus should, come down to kind of above the heart. And yeah, the thyroid is somewhere between the two ends of the thymus, and then the trachea is above it. There's a tube. It's kind of the trachea. So you can see some of the structures more clearly. This is the lungs, right, this side of it. This is the dorsal side of the lungs. In the middle of all is the heart. So we're gonna try to take a look at the brain. So we kind of make a cut around this area. This is where the skull cap is, and then we'll kind of cut off the skull, and then we'll hopefully be able to see the brain inside. Uh, we can see part of the skull. I've cut this side, so not skull, sorry, brain. This is the texture of the brain. Here we have the top of the skull. You can see the brain, at least the ventral side of it. Um, you can kind of see the textures. We're gonna try to get the eye out now, and then maybe we can take a look at it closer. Okay, so here is one of the eyes. Um, we can kind of do an eye dissection on it. So externally, here was kind of the front of the eye. This is the cornea. It's a kind of clear layer that's meant for protecting. When, for example, the animal's eating, say, grass or something, it would be good for the cornea to protect against it. In the back end, we hear this white. Should you see this white portion? That's the optic nerve. This is where kind of the signals that are sent from the eye to the brain in response to like seeing light. You can kind of see it here, this white structure. That's the sclera that kind of surrounds the entire eye. It's white, but right now the entire eye is covered in muscle, so you can't really see the sclera anywhere else. So yeah, we can remove some of the muscles surrounding the eye. This seems to be part of the eyelid still attached, so we can also cut that off. Okay, so then we can kind of cut around the entire eye. Okay, so this is kind of the front portion of the eye. You can, it's a bit dirty, but where's the probe? Notable feature to see, I guess, is the lens. It's this kind of ball or oval-like structure. And when we clear out some of the liquid, you can see the iris surrounding it. Iris it's the structure that's able to kind of stretch or compress the lens, and then that kind of changes how much light is going through. And then if we kind of, I guess, move the eye out, the hole, the hole is kind of known as the pupil, right? And then the pupil kind of leads outward, and then the cornea is over here. The structure, well, not really structure, but kind of the layer, the black layer that's surrounding this entire thing, right? You can kind of see transition, or you can see the sclera and then the black layer in front of this, on top of the sclera. That black layer is a retina. Retina is kind of where the rods and cones are, and that kind of helps the eye detect light. Then the retina will lead out to the optic nerve, which we kind of saw earlier. And so that's kind of how light can enter the eye, and then how the brain is able to determine what the pig's seeing.